Okay, so I've started the recording. Thank you very much, Nico. And I would also like to let you know that the next Cesabinar will be the diverse group. And so it was led by Jos Kefer, Sylvain Glemin, and Andrew Hemstetter. And they will be presenting their work on November 14th at 2 o'clock Paris time. And so now we can start with the presentation, and I will shortly introduce our speaker, Cyril Viol. So Cyril works at the CEF, so it's this Centre d'écologie fonctionnelle évolutive, Center for Functional and Evolutionary Ecology. He was the principal investigator for the Free Working Group, and so his research field is obviously functional diversity. And more specifically, he works on how plant traits translate into organismal performances and looks at processes at community and ecosystem scales as well. All of this is in the context of anthropogenic changes and more specifically land use and climate. And he develops diagnostic and predictive tools for eco and agro ecosystems and their functioning. And yeah, we're very happy to have him present. And Cyril, you're welcome to turn on your video so that people can see you present at the same time as they see your slides. Yeah, thank you. Awesome. I'll leave you to it.
Thanks, Camille. So good afternoon. I'm really happy to give this talk today. So about functional variety. So yes, this came from this free working group. I'm contributing to lead with Lucy Mao, who has been a postdoc of this group for a long time. She's here with me today, and so she will help me to answer your question. So when you think about rarity, you can think of a species or of a plant alone. So maybe it's important, it's precious to conserve because it's quite unique. But maybe this rose is quite common, in fact, compared to other flowers. So is it really interesting, useful to conserve? So this is why we try to introduce a new facet of variety and diversity that we term functional variety. And so this is the title of this talk. So functional variety from detection to conservation. So I guess most of you have tried to look to search on the net pictures of weird things, especially weird animals. So for instance, you can have the longest animal on Earth. You have this pink dolphin and this quite weird bird. So why do we have this kind of weird things? Is it important for the functioning of communities and ecosystems? And this is true for plants, true. And most often we are talking about big stuff like big seeds, big flowers, big trees. And so, for instance, big trees have been searched for kind of trophies, especially in the US. And so, yes, biological diversity is huge, continuous from really small things to really big things, and with the presence of extremes. But it's not so easy to describe this diversity and to describe an organism, a phenotype. It's not just based on a single feature, on a single trait. And often we need several dimensions to describe an organism, the functioning of a phenotype. And this has been discussed quite a while in functional ecology, especially for plants. And so the idea, one solution is to jointly describe several traits, measure several traits to really capture the multidimensionality of the phenotype. And for instance, in plants, we have dimension of stem, flowers, seeds, leaves, and so on, that describe different functions of the plants. And this has been formalized with the concept of functional traits, and we have a non-exhaustive list of features of functional traits that can be related to different plant functions, like, for instance, seed mass, phenology that can be related to dispersal, establishment, and so on. At the leaf level, so the leaf area or leaf dry matter content that is related to leaf metabolism, and so on. And thus the idea of functional ecology and plant comparative ecology is really to use these traits to compare different plant species or different species in general, to classify them, to compare their functioning. So this can be done at the interspecific level, but we can do that also on the right at the intraspecific level by, for instance, comparing different genotype of a single species, here Arabidopsis thaliana. And so this has been done, for instance, at global scale by Sandra Diaz and collaborators by describing the global spectrum of plant form and functions by using a small list of plant functional traits. And so they get this kind of pictures, which is a functional space, or which is a PCA. So each dot is one species based on the comparison here of 6 plant functional traits. And the idea is to look at common features, try to understand different species. And so, for instance, here you have kind of common trends in red by, on the left, more herbs, and on the right, more trees. But on this functional space, you have also, especially at the margin, some extremes, for instance, big trees, but also really small plants with tiny seeds, for instance. So some extremes that can be, for instance, at the margins of this functional space. And these extremes can be important or not. What is quite funny at some point is that here this is at one of the extreme, Arabidopsis thaliana, which has been used as a model species in plant biology for a while. And so the authors said that model species such as Arabidopsis thaliana, located at one extreme of the spectrum, can be positioned against this backdrop, helping to judge how typical or otherwise their physiology and natural history might be. So which means that maybe this is an extreme which might be not so interesting as a model species for all plant species on Earth. Anyway, it's interesting to look at these extremes to try to better understand the whole biological diversity and to try to understand why they are still there in communities and ecosystems. But when we are talking about extremes and outliers, so when you are looking at your Excel sheet, you have big values or really small values. So you are always wondering if it's just measurement errors or kind of ecological extremes. So this is what we try to do. And so we coined these extremes functional outliers. And so this was the beginning of the story of the free working group. So this group was founded in 2018. And so free is for functional variety in ecology and evolution, because we are really asking the emergence and maintenance of functional variety from both ecological and evolutionary perspective. So this group has grown a lot for the last years, and so up to something like 40 researchers, including several postdocs and PhD students who have worked with us. And so the idea of this group was to gather experts in really different fields in ecology and evolution, different subfields of ecology and evolution, and from experts of different organisms. And so the idea was to develop the concept of functional variety, to develop some theoretical predictions, so as to discover patterns at both local, regional, and global scales. And then we looked at some implications for conservation, especially for ecosystem services, which is more or less nature's contribution to people, and also to get some predictions and applied perspective. So we tried to discover patterns on really different organisms who were really open to different databases in plants, microbes, fishes, birds, mammals, and so on. What is important is quite original also is that this working group get funded by both FRB and EDF, so electricity, the electricity company in France. And Anthony Maire, who is part of EDF, is a researcher, is fully part of our group, and we really work with him. He's really a core member of FREE, and so he led some project as part of this working group. I think if you are here, you know what is a synthesis center, but just to let you know that the Cesab, the objectives of this kind of synthesis center is really to work together for a week at least at Cesab, once or twice a year, to really produce a synthesis of current scientific knowledge on a specific topic, to brainstorm on new ideas, to analyze existing data, especially with meta-analysis methodology, but not to produce new data. So it's really important, especially for this concept of functionality, that first we try to explore that with existing data. So based on that, what I will talk today is how to detect and quantify functional variety. What are the causes of functional variety, the implications for ecosystem functioning and services, and how it can help maybe to change to lead to a change of paradigm for conservation. So first, detection and quantification. So when you go to the field or look at a community of plants or fishes, you have a distribution of features like body size or plant height with this kind of distribution. So it's really a classical distribution with a mean value of this trait and also some variation around the mean, which is more or less what we call functional diversity. What is interesting is that distribution can inform you a lot about the structure of communities and the functioning of ecosystems. Especially, you can describe the distribution with the different moments of the distribution, the mean, the variance, but also the skewness or the kurtosis, which inform you about if the distribution is more or less peaky, more or less skewed, uni or multimodal, and so on. And as part of the theoretical work called trait driver theory, we managed to relate the shape of the distribution and so the different moments and different values of the moment of the distribution to different theoretical expectations to explain the response of community to environmental changes and the effect of biodiversity on ecosystem functioning. But then when you do this kind of exercise, so to build this distribution, often you also have some outliers in your distribution. So how to deal with these outliers? So look at this community of fishes. You have maybe a fish species that is quite different from others. So this seems to be an ecological outlier. Something happens. So again, this is what we call functional variety. So how to quantify this? So remember this functional space that you can describe and compare different species in a space, could be a PCA, built based on different functional traits. So how to quantify functional variety is quite easy, at least this is what we propose, is basically to combine, to compute the different distances, the sum of distances with all others. And so we term this index functional distinctiveness, and this is a way to detect and to tell if a species is really different from others. So as part of the free working group, so we build these indices, and also we built with two former PhD students, so Pierre Denel and Mathias Grenier, our package called FunRAR to compute these functional rarity indices and especially functional distinctiveness. So this is based on distances between species, but we can do that also between individuals. And even more recently, we extended this concept to microbial communities. And in that case, we build these indices based on genes instead of species. So it's quite complex, this figure, but just to say that when you have different microbial communities, instead of focusing on taxa, which can be quite difficult in microbial communities, you can directly focus on genes and do exactly the same exercise. And then you can do that across scale, across biological organization levels, but to compare different species and to really target some species that are different from others. But you can also do that at the intraspecific level again by identifying some genotypes that are different from others. And then, again, on this multidimensional space, then if a species is really different from others, like this guy, or an organism is different from others at the intraspecific level, you call it a functional outlier or a functionally rare species or genotype. Maybe this is the most important figure. This could be the take-home message of this talk, is that what is the difference of being abundant or rare or being functionally distinct or not? So this graph is a B-plot. So on the x-axis, this is the abundance of a species. So on the right, this is the rarest species. Okay, just based on frequency, just based on abundance. Each dot is one species, in that case is coral reef fishes. And on the y-axis, this is functional distinctiveness, so on this functional space, if a species is different from all others or not. Okay, so on the top, this is the more functionally original species. And when we explore different data sets for different organisms, almost most of the time we get this kind of relation of lack of relationship. So we found no relationship between rarity in terms of abundances and rarity in terms of traits. So we came with this conceptual framework saying that we can have all the cases. There can be some species that are really scarce, inabundant, but have quite common traits. We can have some species that are dominant, abundant, but have distinct traits. And of course, we can find in ecosystems some species that are really scarce in terms of abundance and also have distinct trait values. So we can imagine a gradient of functional rarity, and maybe, of course, I will go back to this later, but for conservation, it's more important to conserve these guys because they are in danger in terms of abundances, and also because they display distinct trait values. So we thought that functional rarity can be a new facet of diversity and rarity to play with in ecology, evolution, and maybe also for conservation. So if we buy this concept of functional rarity, one first question is that why? Why we still have some functionally rare species, organisms in communities? So this was the main objectives of the work, and we have worked quite a lot to try to answer these questions, where functionally rare species are more likely to occur. We get some hypotheses. We add some hypotheses that it could be in stressful environments that are more adapted to stressful environments, maybe in disturbed environments, maybe under fluctuating environments, and also maybe in marginal habitat of species. So we analyzed several data sets, and I will give you just only one example here, led by Nicolas Loiseau, who was a former postdoc of the group. He analyzed, especially with Nicolas Mouquet, so the global distribution of functionally rare mammals and birds at a global scale. And what he found is that specific hotspot of functionally rare species, in Madagascar, for instance, among others, that were really different from classical hotspots of taxonomic diversity, which means already that in terms of conservation, if you don't have a perfect overlap between taxonomic rarity and functional rarity, well, maybe it's really important to consider this other facet of rarity. So more broadly, to answer the questions where functionally rare species are more likely to occur, you need to trust me because I don't have the time to show you all the results, but as a synthesis, what we found in general is that we get always the main results, which are more or less common to mammals, birds, and plants, is that functionally rare species are more sensitive to stressful conditions, especially current and past temperature, to human footprints, and they are more sensitive to future climate change. So clearly, they are more endangered than common species. So this is at large biogeographical scale. But what's happened within a community? So within a community, we have this classical figure in community ecology with some species that are dominant and others that are scarce in the community. But we have also this with the presence of some functionally distinct species compared to others. And this is a key question for us and for community ecology. Why do we have the maintenance of these extreme phenotypes in communities? And again, I insist on the fact that this is not necessarily the dominant species that are distinct in communities and vice versa. So this is a new question. Why do we have these extreme phenotypes within communities? So it's not so easy to answer. And for that, what we did is a theoretical synthesis to try to really recast some long-lasting hypotheses in community ecology about the maintenance of rare species in communities by recasting them in terms of phenotypic traits. So this result has been published in this paper led by François Munoz about the ecological causes of functional distinctiveness in communities. And one reason, among others, is that we can still have a different peak of fitness within the communities, and that some combination of traits that are quite original can be maintained based on the fact that they are adapted to really local conditions, but on a secondary fitness peak. So this is something that we are trying to test that with data, but it's not so easy to test because we need a lot of data and individual data to test that. So next, we ask why, and now what are the consequences? If this functional distinct species exist in communities, what could be their role at the ecosystem level? So part 3, the role in the regulation of ecosystem functioning and services. So I guess you are all really very well aware of this kind of literature and results in ecology, especially a subfield of ecology which called the biodiversity ecosystem functioning relationship. So in the 90s and after, there have been a lot of theoretical works and experiments also to test the effect of biodiversity on ecosystem functioning. So what is the effect of the number of species in an ecosystem on ecosystem functions like ecosystem productivity? And a lot of you, I guess, know this kind of relationship with a positive but saturating relationship between the number of species and ecosystem productivity. But this is just based on numbers. So the idea of this graph is that you add one species and you increase your productivity, which could be quite naive to imagine, but this is what's happened from experiments. Without really considering the different functions that the species can display. But of course, in a community, you sample different species that are different based on their plant functional traits, probably because they play different functions in the ecosystems. This is true for plants, fishes, and other communities for sure. So how to consider that and to use that to really to better understand the effect of biodiversity on ecosystem functioning and maybe to better predict variation in ecosystem function. So one key tenet of functional ecology is that traits and the trait distribution within community matters for that. So the community level trait distribution can be a key driver of ecosystem functioning. And this is exactly what the trait driver theory predicts among other theories. Especially the mean of the distribution, but also the variance can be key. Especially the mean, because the mean, the average value of the trait distribution within a community could reflect the effect of dominant species, which has been termed the mass ratio hypothesis in functional ecology. But also the variance of this distribution can be important because it relates probably to the effect of niche complementarity among species within the community. So there have been plenty papers and syntheses testing that, especially in plants. So especially in this book by Eric Garnier and Marie-Laure Navas in French and in English, they produce a synthesis showing that yes, trait distribution matters, especially community weighted mean are the key drivers of several ecosystem functions. But what about these extremes? Is it important or not for the regulation of ecosystem functioning? So this is what we try to test in this working group by looking at the different moments of the trait distribution, but also this extreme phenotype. We did that at different scale, especially at the scale of the Alps, with one postdoc, Philippe Brun, who used data from more than 45,000 botanical relevaes in the Alps, for which you have the species composition of all these communities, but also the productivity of these communities thanks to remote sensing data. So the first thing we try to test is the effect of species number. In indeed, again, in theory, we expect a positive relationship between the number of species and ecosystem productivity. But in our analysis, we found a flat relationship. In practice, no effect of species diversity at all on ecosystem productivity assessed by remote sensing. Then, we tried to test the effect of the different moments of the trait distribution, so the mean, the variance, and also the effect of specific species, probably maybe with different, specific trait values. And so here is the results. So this is a summary of model selection. So on the y-axis, this is just a percentage of explanation of deviance. And on the y-axis, this is the different moments of the trait distribution for three traits, SLA, leaf nitrogen content, and height. So the mean, the variance, the skewness, and the kurtosis. But also the effect of key species. So some species, thanks to a specific algorithms, we try to identify beyond species diversity, beyond these community-wide drivers, we try to test the effect of the presence of key species in the regulation of ecosystem productivity. And so what we found is that, yes, the trait community level trait distribution matters. All the moments are important to explain variation in ecosystem productivity. But most importantly, this was the presence of some species that really drive variation in ecosystem productivity at this scale. Then we try to look at the species and who are they on the functional space. So always the same thing in a multi-dimensional space based on these three traits. We found that these species are quite specific, not extreme, but quite specific. Some were legumes, but not only. And this is these species that really drive ecosystem productivity beyond species number, beyond community level trait distribution. So one of the messages of this study is that species number is not enough. Trait distribution is probably not enough. And this is really the effect of some species, maybe with some specific trait combinations that explain most of ecosystem variation. But we still had this idea in mind that maybe really rare phenotypes are really more important than common ones to drive ecosystem functions. So our idea, so this is this conceptual framework on the right, is that think of a biodiversity erosion scenario. If you are losing randomly some species, you may have a decrease, more or less linear decrease in a given ecosystem property. If you are losing a common species first, in blue, maybe this decrease will be less important, at least at the beginning. But if you are losing functionally distinct species first, because there are specific functions in the ecosystems, the reduction, the decrease in ecosystem function will be more drastic, especially at the beginning. So this is some hypothetical cases that is really difficult to test, especially without experiments. Especially because we are not allowed to run, to perform experiments in the case of Cesab project, but we will do it at some point anyway. So what we did is a kind of experiments, virtual experiments with modeling, with mechanistic modeling, with forest data. And so for that, we used a forest gap model, the forceps model, developed by Xavier Morin at CNRS. He developed it to model ecosystem dynamics and functioning. And he has really a mechanistic relationship between threats, and he has a lot of threats, and we can create artificial ecosystem forests with more or less species, with different threats or not, and look at the consequences for ecosystem functioning, especially productivity, after 100 or even more, in our case, 2,000 years. So we did that with one of our PhD students, Léo Delalande. And so we specifically test this conceptual framework by performing a lot of simulations to do all the possible cases, by starting with a community of 30 species, removing them one by one randomly, or removing one by one, but starting with the most distinct species in red, or the most common species in blue. And here is one of the results. So on the x-axis, this is the number of species removed in these virtual experiments. So from 1 to 30. On the y-axis, this is the total biomass production of the forest. In gray, this is all the random simulation when we are removing one by one species, but without accounting for their threats. The red line, this is when we are removing one by one, but the most functionally distinct species first, and so on. And in blue, this is when we are removing the most common species first, and so on. And we exactly validated our expectation, at least in this specific site, that ecosystem function, in that case total biomass production, drastically decrease when we removed the most distinct species first, and not in the case of the most common species first. Which means that, at least for the long-term ecosystem functioning, ecosystem productivity, yes, probably functionally distinct species matters for the regulation of this function. Well, this is clearly not enough in terms of demonstration, and we are now working more on different data sets, but also on experiments within and between species to try to really understand the role of rare phenotype and for different ecosystem functions. And if you have other data sets in mind, we will be really happy to consider them, because it would be interesting really to test that. It's really important to somehow to validate our concept, to try to see whether and how really functionally rarity matters for ecosystem functioning. So if we think that, yes, it's an interesting facet of functional rarity, is an interesting facet of diversity and rarity to consider for ecology and evolution, maybe this has some consequences for conservation also. So this is part 4, a change of paradigm for conservation. So there is a very long-living debate in conservation biology whether to conserve species because they are in danger, because they are scarce, or ecosystems because we need them, and especially several services to save as a kind of natural capital for us. So this is really two opposite point of view in ecology, and especially in conservation biology. As you can imagine, this has been criticized a lot, especially the ecosystem level, because this is a way to put some money, to put a price to nature, to ecosystem services. So a lot of people have criticized this neoliberalist view of nature. And also some people criticize this, especially the fact that, well, the ecosystem level is kind of buzzword, but it means nothing, in fact, for conservation that should focus on the extinction of species, not ecosystems. Maybe, but at the same time, there are urgent needs in conservation biology. First, there are too much species to evaluate for their conservation status. So we need to speed up this evolution, maybe with some rules to do that. There are probably also, but not probably, there are too much species to protect. And so we won't be able to protect all. So a triage of species is needed. Maybe we have some species that we should protect in priority. There is also at global scale the objective of protecting 30% of land and sea by 2030. So this is the coming Montreal Global Biodiversity Framework. So, but to do that, where to locate protection areas. And finally, it's really important probably to protect biodiversity and ecosystems for us, because this is the basis of what we call nature's contribution to people. But so how to do that? So we argue as part of TREAT that threats can reconcile both targets of conservation. This is a kind of en même temps strategy, maybe, which means that threats at the species level, but also at the community level, can be good proxies for biodiversity, ecosystem functions, but also the contributions of nature to people, so NCP, which is the new way to consider ecosystem services. And I don't know if you are familiar with these nature contributions to people, but one way to illustrate them is to say that you have three different kinds of nature contribution to people, at least in this nature for future framework, which is nature for itself on the top, on the left, nature as culture, living in harmony with nature, and nature for society on the right, so the benefits of nature to people, which are the former ecosystem services. And when we review the literature on threats, in fact, threat can be important for this free summit of this nature's contribution to people. So nature for nature. So here is an example with fish communities. So at the community level, when looking at community level threat distribution, especially community level threat redundancy, is a good proxy of community sensitivity to disturbance. Of course, there are many, many examples that threats are a good proxy of ecosystem services. For instance, in that case, this is for plants that community mean SLA, so the leaf specific leaf area, so one dot is one community, is a good proxy of biomass production of these ecosystems. And for nature as culture, we start to have some examples also that threats can be a good way to evaluate them. So here, this is an example at the species level for mammals. So on the y-axis, this is an index of charisma of the species. And on the x-axis, this is functional distinctiveness. And we found quite a strong correlation between this charisma index and functional distinctiveness, which means that threats could help to quantify this nature as culture contribution. So based on that, we said that traits can really help to inform conservation targets, especially because when we try to compare the distribution of taxonomic rarity, taxonomic diversity, and functional rarity, we have different hotspots for all organisms we were looking for. Here, this is an example again with mammals and birds. So on the top, this is the distribution of the number of species. And on the bottom, this is the distribution of functionally distinct species. We have different hotspots at global scales, but also at regional and local scales, which means that we are probably missing something if we don't take into account these facets of rarity in conservation. Of course, and this will be my conclusion, so there are lots of limits and perspectives. Even if we have worked quite long on this concept of functional rarity, we are still missing a lot of things. Especially when I presented you these pictures, I said that they are really weird. They are weird because they are, for instance, this guy is the longest animal on Earth, so we can capture it with some traits. But for others, for instance, these weird birds, so the description is that this is a stink bird due to their foul odor, which is caused by the fermentation of food in their digestive system. Clearly, this is the kind of things it's really difficult to capture with traits because we cannot really a priori measure this kind of traits without a priori knowledge about this ecological originality. So really capturing weird species that can play key role in communities and ecosystems is probably important, but we are far from really be able to quantify it with our concept of functional rarity, especially with the database we are working with. So what's next? Well, everything remains to be done, clearly. Probably we need experiments, sensu lato, maybe modeling, to really test the effect of functionally rare species, especially on ecosystem functions. I think, especially in plants, but not only, more ecophysiological knowledge is needed. And for that, at the end, we need really a thorough evaluation of the role of functional rarity for conservation before really be able to guide a new conservation program. And just to finish, I really want to thank the Cesab and all the staff of the Cesab, which is really a functionally distinct and unique place to work. And so, yeah, thanks a lot.
Awesome. Thank you very much, Cyril, for this very nice presentation. So now there's going to be some time for questions. We're going to spend maybe 15 to 20 minutes for this. And so please, those of you who have questions, please raise your hands using the raising hand tool here in Zoom. And we will enable your possibility of putting your camera. So it would be nice if you have a question to enable your video so that we can see who is talking. It's just more, it's just nicer. So yeah, does anybody have a question? And also those of you who cannot talk, feel free to write some questions in the chat. We will make sure that we check them as well and ask them for you.
well, can I break the ice?
Yes, you can. Nico, go ahead.
I'll do that. I'll do that. So, well, you've been talking about the challenges about, conserving functional rarity, and what is your feeling about the future of functional diversity? I mean, of rare functional diversity. Is it on the decline, given that very rare species in abundance are more likely to be hit by the global change?
So it's not easy to answer these questions because I'm not sure we have a complete evaluation of functionally rare stuff as I said, based on our analysis and with the threats we are playing with. The answer is yes, because they are more sensitive to extinction, probably because of their size, among others. So I would say that yes, in that case. Even if functional rarity for me also relate to ecological specializations. And in that case, it's more tricky to answer these questions because we do not have a thorough evaluation of ecological specialization in that case. But based on the threats we are playing with, the data sets we are playing with, the answer is yes.
There is a
Okay, so
question.
yeah, we have a question from Jesus who raised his hand. So if Jesus wants to, yeah, I invite you to enable your video if you can. If not, we'll just listen to you.
Thank you. It doesn't seem that the video works, but I can just talk it. So I thank you, Cyril, for the nice presentation, and this kind of is refreshing to see something so new as well at the same time. So my question here is, if you, I know it's difficult, but perhaps you can elaborate a bit more about the possible mechanism behind this effect of this rarity. For instance, when you talk about ecosystem functioning and the example of the biomass. So I would understand the species with the most abundant traits perhaps impacted a lot of this biomass because the strength in this case is in the numbers, right? What do you think is the mechanism behind this rarity and the effect on the functioning?
Well, this is a very good question. So in the case of the modeling exercise I showed you, in fact, this was indirect effect based on the fact that the most distinct species were the species the most adapted to local conditions. So they played a key role in biomass production because they were more adapted to stressful conditions in our modeling exercise. So it was not directly an effect threats, but more response threats based on their adaptation to local conditions. But we can imagine also that on the long term, these functionally distinct species are more likely to contribute to ecosystem biomass production because they are involved in regeneration processes and so on. But it's really difficult to really, even with a model for which we have all the mechanistic relationship, it's really difficult to identify the mechanisms behind this.
Thank you, Cyril.
Thanks for your question. Now maybe we can have Guillaume's question.
Hello, Cyril. Thank you very much for the presentation. It was very nice. There was quite some work also on phylogenetic distinctiveness and branch length of rare clades. Are these two things correlated? So you could expect that phylogenetically distinct clades might have evolved for a longer time, very distinct sets of traits. Do you observe that?
So in terms of correlations, it was not so clear, especially in our 2017 tree paper. We had not really weak relationship between evolutionary distinctiveness and functional distinctiveness. So it's probably not enough, but there is a huge debate whether phylogeny is enough to capture traits or not. So probably in a multi-dimensional space, when you have, it can capture more or less trait diversity, but in the case of functional distinctiveness, we get a weak relationship between evolutionary distinctiveness and functional distinctiveness. And more specifically, in the case of the effect of functional distinctiveness on ecosystem functioning, so you can expect, of course, based on adaptation and so on, that we have a conservatisms of niches and then of traits for response trait, but for the traits that are involved in ecosystem functioning, we call them effect traits in functional ecology. It's not so clear that we have a strong conservatisms of these traits. So if we are really directly focusing on functional distinctiveness instead of evolutionary distinctiveness, it could be more important, especially in the case of ecosystem functioning.
Thank you.
Okay, thank you very much. Maybe a question from Bertrand, and then we have questions in the chat that I will ask. But Bertrand, you go ahead.
Okay, thanks a lot from my side for the very nice talk. I was just wondering, how can you integrate actually the concept you were mentioning about functional rarity at the trait syndrome level? Or how can we integrate correlation among traits into functional rarity?
So it's part of this because the way we are quantifying functional rarity is a combination of traits. So it's a syndrome of traits. So in fact, they are pinpointing really original trait combinations, trait syndromes. And when I showed the PCA of Sandra Diaz and collaborators, we have this red hotspot in the functional space that are common trait syndromes, but we have also original trait combinations, and we can identify them directly based on our functional distinctiveness indices. So for me, it's totally related. I'm not sure I answer your question, Bertrand, but...
If I may complement, you might have different variability in correlated traits like SLA and whichever others.
Okay.
Maybe you can be rare for SLA, but not for whichever other correlated traits. That's basically the idea.
Well, that's a good question. So we started to do that, in fact, at the intraspecific level with Arabidopsis. And in fact, indeed, functionally distinct genotype have different trait correlation. So maybe the other evolutionary pressures act on them. So yeah, this is really the beginning, but it's really important that, interesting and intriguing that indeed functionally rare things maybe are under different evolutionary pressures and selection pressure, sorry. And so, yeah, that's only the beginning, but this is exactly what we observe at the intraspecific level. So this is really something we want to look at more.
Okay, thank you. I am going to read out a question from the chat. So it's a question from François Massel, and he says, traits are also very likely to be correlated. For example, animal traits linked to overall body size or body shape. How does the number of retained traits affect measures of functional rarity?
So of course, in all our papers, we perform sensitivity analysis because always the reviewer asks us, what about the number of traits? What about the identity of traits? And based on several reasons, including mathematical reasons, there is a tight relationship between functionality and whatever the identity of trait. It's not so true, but yeah, this is more or less based on the way functional spaces are built, that when you have enough threads, 4, 5 threads, you have a strong correlation between functionality, the functional distinctiveness when you put more threads. I'm not really proud of this answer, but this is what's happened mathematically.
There's another question in the chat by Boldorini. He says, thanks for your presentation, Cyril. If I understand well, the functional rarity concept was applied to affect traits linking to ecosystem functioning. But I would like to know if we can apply functional rarity framework to response traits. For example, to see if functional rarity species are more or less affected to biotic or abiotic threats than common functional species.
Well, we clearly did both in 3, and the part 2 of my talk was clearly on response threats. So all our works on the distribution of functionality at both local and large scale was the response threats. Somehow, even if we do not have always a hypothesis about this response threat, so clearly it can be and it is applied for response threats. And our ecology letters papers on the causes of functional distinctiveness in communities was clearly based on response threats. And what we found indeed is that functionally distinct species based on response threat were more likely to disappear in the future. So.
Okay, thank you. Maybe we can take this last question from Samuel, and then I have a few questions about leading a group, a Cesab group, and its functioning. But please, Samuel, if you want to ask your question, go ahead.
Yeah, thank you for your nice presentation. I was wondering what is about exotic species if you carry on your reasoning at the limits. To add exotic species is probably, add some complexity to the ecosystem and some rarity also. And so would the conclusion be that it is the functioning of the ecosystem is better than?
But first, we performed some analysis, and yes, it seems that exotic species or invasive species are more distinct, so they are the margin. So it's true. And one consequences that yes, for some ecosystem functions, ecosystem function could be improved, but it's hard to say like that. I don't know if you have a strong opinion about that. For productivity, it has been shown. But of course, we also know that invasive species have strong negative impacts on ecosystem functions. I would be a bit careful.
Thank you.
Okay, so then I would also like to ask a question, and it's more about, the functioning of a Cesab group in general. So you were the main coordinator of this free group. It's quite a big and dynamic group. And I think that here in the audience, there might be quite a lot of other members of Cesab groups, and some of them have just started. And I was wondering whether you could share some tips with us all about, I don't know, like what are the successful things that you managed to put in place to have a nice dynamic in the group? Or on the contrary, are there some pitfalls or some things that, that now with your experience, you could share with us so that we don't fall in the same traps as you did when you started? And also, I don't know, I see that Lucy is here and maybe has experience as a postdoc within the group, maybe she has some insight to share. Anyway, this is a dynamic discussion. Please feel free to share.
Well, maybe I will start and see. I'm not sure I have good tricks for leading this kind of group, at least be flexible. And this is what I and we like at Cesab, is that it's really flexible in the functioning. It's really third, the Cesab is really a nice place to work and to live. So people likes to come, and so it's really nice to be at Cesab and really to work all together. The idea is to be quite flexible also in terms of the people we have invited for the whole duration of the project. And what is important, at least for us, what has been important is to involve our postdocs and PhD students in our group as much as possible, because they are really doing the work. And it's a win-win. It's for them. I think it's great to be incorporated in this group. And yeah, it has really helped to spread our concept and to really test it with different databases and so on. What is more difficult for me personally is really to continue interactions between meetings. So it's not so
Yeah,
easy.
exactly. Like, how do you keep the momentum going on? Because as in many projects, everybody's very enthusiastic
we
in
had
the
a lot of
beginning.
projects, and yeah, the group is really big now, so it's always difficult to put some pressures on people. I don't want to do that, and it's not so easy. So I'm not sure I have a good advice.
Well, I think we can say that you were successful because the dynamics and actually the science, I don't know, productivity of the group was really good. So
yeah,
it's been
the
going on
idea is
for a
that
while.
people are happy at least during the meetings, and then in between, yeah, it's more difficult to keep the dynamics.
Okay, so no whipping then. Okay, I thought maybe you would tell us that. Lucy, do you have any insight you want to share?
Yes, thanks a lot, Camille. I think from a backstage perspective, a key point to have a successful week meeting at Cesab, I think it's the organization before the week happens. And Cyril didn't mention it, but he spent, and now we spent quite a long time to organize the schedule of the week, trying to organize who is going to speak, to present which topic, to have the best time schedule possible during the week, to have a good balance between novel project presentation, update about long issue project sometimes. So yeah, the amount of work before the week is not negligible at all, and also with you guys from the Cesab to have all the organization for the dinner, etc. So I think it is one key point to have a successful week. And then as a postdoc, for me, with the free working group, which is very maybe special because it is a long time history, it's just super nice. It's a super opportunity because you have all these dynamics which is already in, with a novel concept to play with. And so it's very nice and easy to test many different things. So yeah, for a postdoc, it's just a huge opportunity, and I hope I will. I am pretty sure that free gives me a lot, and I hope I can give a little bit to the free working group at the end also.
Well, sure you do. And it's true that also what you were saying about organizing the schedule. I know that for people that are working here at the Cesab, it really helps to have this schedule as well, so that we can know when you're doing your, like, what are the different topics during the week, and so that people and the other postdocs that are here at Cesab can join in. So yeah, I think I agree that that's an important tip. Is there anything else? Are there other questions maybe in the chat? Does anybody else have any questions they want to ask before we end this session? Nico, did you want to say something maybe?
Well, I mean, I just want to thank you all. I mean, we've been reaching our carrying capacity today of 100 people, and we're gonna, because of you, Cyril, we're gonna have to update our Zoom lessons for hosting more more people for the next meeting. So thank you very much, Cyril, and you've been just doing a great presentation, and thank you all for this attendance and interesting question session. Thank you.
Yep, indeed. Thank you very much. And yeah, so keep posted for the next Cesabinar, and the recording of this session will be put online on the FRB's YouTube channel, maybe in a week or something. And so I will stop the recording now. Thank you very much. Goodbye, and see you next time, hopefully.